Everyone, welcome back to our channel. Today, we have a fascinating experiment in store for you. We'll be demonstrating how to extract DNA from blood using basic laboratory equipment. It's an exciting process, so let's get started. Here are the items you'll need for this DNA extraction experiment. A blood sample, you can use your own or obtain it from a medical professional. A test tube, a centrifuge, distilled water, a tube rack, a pipette, a tube of lysis buffer, a tube of protease K, a tube of isopropanol, and a small glass container. Safety precautions should be followed, such as wearing gloves and handling blood with care. To begin, we'll start by taking a blood sample. It's important to note that handling blood requires caution, so make sure to follow proper safety procedures or consult a medical professional if needed. Now that we have our blood sample in the test tube, we'll add the lysis buffer. The lysis buffer helps break open the blood cells and release the DNA. Next, we'll add a few drops of protease K to the test tube. Protease K is an enzyme that helps break down the proteins in the blood, allowing us to isolate the DNA. After adding the protease K, we'll gently mix the contents of the test tube to ensure the enzymes are distributed evenly. Now, we'll place the test tube in the centrifuge. The centrifuge will spin the mixture at high speeds, separating the DNA from other cellular components. While the centrifuge is running, we'll wait for the spinning process to separate the DNA from the rest of the components. Great! The centrifugation process is complete, and we can see that the DNA has separated into a pellet at the bottom of the test tube. Now, we'll carefully pour off the liquid portion, known as the supernatant, leaving behind the DNA pellet. To wash the DNA pellet and remove any remaining impurities, we'll add a small amount of distilled water to the test tube. After adding the water, we'll gently swirl the test tube to mix the water with the DNA pellet, ensuring it is properly washed. Now, we'll place the test tube back in the centrifuge for a quick spin. This will collect the washed DNA at the bottom of the tube.